Hi everyone, this lesson's on forensic entomology. Let's say a body was discovered in the woods. The time and cause of death are unknown. The body appears bloated and smells really bad, and small insects like you can see here have been found on the body. Could you determine how long that body's been there? Let's find out how. What can insects tell us when analyzing a crime scene, particularly when a body is discovered in an outdoor environment? Well, quite a lot. It can reveal the location of a body if I see insects going towards it. It can determine if a body was moved based on what kind of insects I find and how long they are into their development. It can identify the geographic range of a crime, as insects are specific to certain regions of the world. It can link a suspect to a crime scene or to a victim based on insect materials that may have been passed along. It can indicate the travel of a suspect and trace the origin of drugs, because insects are animals. They eat. They may be eating substances or tissues that contain substances such as drugs in them. So entomology is what we refer to as the study of insects. Insects are defined as having three unique body parts or segments. First part is the head. That's where you find the sense organs, the eyes, the antennae, and the mouth. Then there's the thorax right below the head, and below that is the abdomen. Insects have an exoskeleton, which is just an external skeleton. The exoskeleton does not grow, so as the insect grows inside the skeleton, it has to shed or molt its exoskeleton and grow a new one anytime it gets bigger. Most insects have wings, but not all. And each species prefer a specific location, temperature, and food source. Because of the specificity necessary for their survival, this is part of what makes them perfect in forensic investigations. They have unique geographies, and we need to find unique geographies in solving a case. An insect's life cycle, generally speaking, typically follows 4 phases through a process called metamorphosis. Metamorphosis is the transformation in form from a youth to an adult. Let's look at the generic life cycle. Most insects start as an egg. The egg is laid by a female, typically in some kind of decomposing matter or some food source. Those eggs will then hatch into larvae. Larvae are able to move, and they are hungry. They eat again and again and again. If you've heard the phrase maggot, a maggot is just the word for fly larva. Since flies are very common, so too are maggots. As those larvae eat more and more nutrients, they'll develop into a pupa. A pupa is a non-eating subadult. This is where that larva is going to change and develop into the adult. Some mature form, typically winged, but not always. Most insects follow this 4-step process, but there are deviations in the theme based on the species you're working with. With insects, it's all about location. Insect species live in unique and specific niches. A reminder, a niche is the unique temperature, precipitation, and behaviors that a species needs to survive. Because of the competitive exclusion principle, no 2 species can occupy the same niche at the same time. So if I find an insect on a body or on a suspect, I can conclusively determine what niche that insect must have come from and what geographic location it came from. Insects vary across geographic locations, so we can use them to track where a suspect or a body is or may have been. This is why discovering them is so informative. And by understanding their life cycle, we can get an understanding of time. We know how long it takes for each phase in an insect's life cycle. So if I find eggs, for example, that body's probably been there a lot shorter time than if I find adults. Insects and decomposition go together very well. As living tissue decays, that living tissue is going to begin to produce different temperatures, nutrients, and niches. As this progresses through, different insects will interact with decomposing matter at different times and locations, because as that object decays, it's providing unique niches. This is going to enable forensic investigators to determine where on the body injuries may have occurred. Insects are likely to be attracted to an injury because it's exposing tissues and fluids that they'll use to reproduce. And you can determine if a victim was restrained. When you're restrained for a long period of time, your body's likely going to have urine or fecal matter on it. Those are 2 substances that certain insects are attracted to, something we can track. And was the body buried or submerged? That's going to determine what insects can reach it and which ones can't. So if I find a certain type of burrowing insect that I know likes decaying matter, there might be a body buried nearby. And also, what was their exposure to toxic chemicals or drugs? As insects eat decaying matter, they're going to also be eating any of the substances such as drugs that are in that body. So let's first understand decomposition before looking how insects interplay at each phase. There are 5 phases of decomposition we want to know. I'm going to go through each one. First phase or stage of decomposition is the fresh stage. The fresh stage is 1 to 2 days after death. During this phase, the pH of the organism is going to lower and cells are going to begin to break down, something we call autolysis. The face is going to begin to become discolored, and flies will begin to lay eggs in openings, typically mouth, eyes, anus. In the second phase, we're looking at 3 to 4 days after death. This is the bloating stage. In the bloating stage, the body becomes bloated. It starts to decay, so we have putrefaction, we have decay. Also, the abdomen's going to begin to swell or bloat, and it's going to swell with CO2. The reason for this is the bacteria inside the intestines begin eating the dead organism. All of that metabolism releases a massive amount of CO2 inside the abdomen of the organism that died. That bloat will continue until it bursts. The skin will begin to marble, and the temperature of the corpse is actually going to increase because of all that bacterial action in the gut. So we had the fresh stage, bloating phase. Now we have the active decay stage. The active decay stage is 5 to 10 days after death. The bloat continues and we get an eventual burst where the organism's remaining tissues just can't hold back all that CO2 anymore. And yes, it makes a sound. We'll learn more about that when we look at body farms. The tissue on the organism liquefies and the skin begins to fall off. You're going to have a massive loss of body mass during this time because this is the highest point of insect activity. Multiple different insects are going to be eating the body and assuming that mass. Especially maggots. Maggots and larvae, like we saw with the larval stage, eat and eat and eat. Beetles often arrive at this phase as well. Beetles don't come in first, flies always do, but beetles will arrive later and feast on the decaying corpse. After active decay is advanced decay. This is 10 to 24 days after death. The body dries and begins to preserve. Most of the flesh has gone away, so very few insects remain. Only larger insects like beetles who have the mandibles, the mouth parts to be able to pick away off the bone. The last phase is the dry phase. This is 25 plus days after death. At this point, only bones and hair remain, so there are very few insects. There isn't much for them in way of a meal. And this phase can take weeks or years depending on the environmental conditions the body is in. It all depends on the climate. So each of these stages of decomposition actually attracts a unique insect species. We call this series of events insect succession. Insects change or metamorphize over a predictable time frame. So if eggs start during the fresh phase, I can accurately predict what I would find during the active or advanced decomposition in terms of the insect's development. Knowing the timeline and changes of insects attracted to corpses can provide this timeline. Let's look at an example. So here I have a fresh corpse, so first couple of days. Blowflies are typically the first insect to arrive on a corpse. Because they can smell a dead body from about a mile away, they'll get there within minutes of the organism dying or appearing at the crime scene. And their life cycle is very predictable. We know that soon after death, the adult's going to land near an opening, probably a mouth or eye, and lay its eggs. Those eggs will develop, and in about 1.8 days, they'll hatch into a larva, which we're going to call an instar 1. As larvae develop, they become bigger and they have some internal changes. So we're going to refer to them, developmental phases of the larva, as instars. It's just a series of events going from small to big. So after these tiny instar 1s develop, we get to about 2.5 days and they'll grow into an instar 2. You can see in the diagram they're just a little bit bigger. At 4 to 5 days, they'll grow into instar 3s, very, very large larvae. And 6 to 12, they'll be in a prepupa stage. They're beginning to form into pupas. 18 to 24 days, they are a fully formed pupa. And 21 to 24 days, we get an adult blowfly. This gives us an incredible timeline. Knowing that blowflies arrive at the corpse right when they are present or right when they decay, if I find some prepupa, corpse is probably 6 to 12 days old. Let's look at everything lined up at once. So here are 5 stages of decomposition: fresh, bloating, active decay, advanced decay, and dry remains, and the time span for each. Let's put that on top of the life cycle of the blowfly. In 1 to 2 days, if I should find eggs and instar 1 larva. In 3 to 4 days, I should find instar 2 and 3 larvae. In 5 to 10 days, during the active decay phase, I should find some prepupa and instar 3. During advanced decay, I should find some prepupa, pupa, and maybe near the end some adults. And at 25 days, I will find an adult. So if I find a corpse and I'm not sure which phase of decomposition it's in or how long it's been there, and I find some instar 2s and 3s, odds are that body's been there 3 to 4 days. It's not 100% accurate, but it's a pretty robust estimate of how long a body's been present and how long it's been decaying. So at the fresh phase, we have our blowflies like I show you, but also flesh flies will arrive as well. These are flies that feed on flesh. So if a body is exposed to the elements, you'll probably find flesh flies, but if a body is wrapped or covered, flesh flies can't find the flesh, so they probably won't be present. Yellow jackets arrive during this phase as well. Now, moving on to the bloating phase, we still have blowflies, flesh flies, and yellow jackets, but some new insects come in. We'll get some house flies, some coffin flies, which are the tiniest and the only ones that can actually get into covered bodies. They're small enough to fit through the fibers of a cloth. And beetles will begin to arrive to eat the bloating corpse. We'll also get to see the development of maggots, of larvae. So if you see maggots or larvae, that's a clear indicator that whatever the maggots are in has probably been around a couple days. A couple notes about beetles. Their life cycle is very, very similar to flies. They go from eggs to larva to pupa to adult, but there are some subtle differences. One is their larvae are called grubs because they have legs. This enables them to move around. If you have any reptiles that you feed mealworms, a mealworm is a grub. They arrive later than flies because they're going to be mating on the corpse. And adults have 2 sets of wings. Flies only have 1. With the beetle, 1 set is for flying and the other set is just for protecting the abdomen. Now let's go back to our next phase, active decay. During active decay, I still have a lot of flies, I still have a lot of beetles, I have some maggots, yellow jackets have left, they're no longer interested. But this is a point where roaches will begin to enter onto the corpse. When we get to advanced decay, don't really have any roaches. We still have some flies, some beetles, but we'll get some fruit flies and gnats that arrive later on in the decomposition process. And with our last phase, the dry remains, we'll still have some flies and some beetles. But this is when you get other organisms. You'll get ants and you'll get arachnids, so spiders and scorpions. Why did I go through what insects are in each phase? Well, as an investigator, what insects you find can immediately tell you what the stage of decomposition is. And since we know the timeline for that, that can tell us how long that corpse has been present. Now, there are a lot of factors that affect this series of events of insect succession, how insects develop and when they arrive. Big one is temperature in the environment. The greater the temperature, the faster the development of the insects through their life cycle. That's why it's important to know what the weather conditions are wherever you find a body, because that will change how you interpret your insect evidence. Also, is the body covered? Because if the body's covered, that's going to keep a lot of insects off of it. You're only really going to find coffin flies that are able to fit in until that cloth breaks down. Also, if there are a lot of predators of insects nearby, that'll manipulate your data. If something's preying on all the flies and the beetles before you're able to collect them, the low number of insects might unintentionally make you think that a body is fresher than it actually is, because a predator like a praying mantis gobbled what would have been a good sample. And the time of day is crucial. A lot of insects will only grow in the presence of daylight. And in terms of their reproduction, their oviposition or their egg-laying behavior, for a lot of them only happens during the day. They won't lay eggs at night. So that can make you estimate a body to be a little younger or older than it actually is, based on if it was placed during the day or at night. Now, larvae can tell you more than you think. Not only can they tell you the potential age of a corpse, when they develop through each instar, they develop a structure called a spiracle. And these are breathing slits. Notice in the diagram how they change from one instar to another. The first instar has kind of a U-shaped spiracle, second has 2, and the third has 3. It's just a quick way to ID how old a larva is or how far in the instar process it is. And since maggots are eating away and eating away and eating away, think about what they're eating. They're eating a body. So if I blend out those maggots together and make what's commonly called a maggot milkshake, I can actually analyze the content of what they've eaten for DNA, drugs, and toxins and poisons. That can be very helpful in determining the cause of death for a body you find. And if we can't tell from a body who it belongs to, this may be the only source for DNA if the body's too far gone. So how do we know how long it takes for each insect species to develop? I've been giving you pretty accurate numbers. It's good to know how we got them. We figured this out determining something called accumulated degree hours, or ADH. It's a number of hours at a specific temperature for a specific species to develop one stage. So for example, the time it takes to go from eggs to larva. Because there are so many different environments out in the wild, desert, rainforests, taigas, and there are so many diverse species, we need to calculate the ADH for every single species in every single environment for each condition. That's a lot of math. Here's how you do it. If I want to scientifically determine the ADH, I just need to recreate the conditions I need to understand. I need to grow insects to full life cycle in the same temperature and conditions that I find at the crime scene. And then each day record the temperature, humidity, and the life development of the insect. So here's some data from such an experiment. You can see going from egg to first instar, what the temperature was, and how long it took. Here's the equation for getting ADH. You need to determine the adjusted average temperature and multiply it by the number of hours it took to go from one phase to another. First step is just calculate an average temperature. Each day the temperature is going to fluctuate. So take the lowest and the highest temperature for the day, divide it by 2. We need to get the adjusted average though, and not just the average present. And here's why. A lot of insects have what's called the lower limit threshold. There's a temperature to where their metabolism shuts down and they cease to be. We need to account for that and adjust our numbers accordingly. So to get the adjusted average, you're going to calculate the lower limit, it's typically 10 degrees for most insects, and subtract it from the average you got in the lab. After doing that, you then just multiply by the number of hours, and now you have your ADH for each phase. So you can see here for the egg, we had 23 hours at 70 degrees Fahrenheit, multiplied that together for an ADH of 1,610. If we want to know the full life cycle of an insect, what we call the cumulative ADH, just take the sum of all the ADHs. This can be used to determine the PMI, to determine the time of death. It's not 100% accurate, but it's helpful in determining an estimation. When processing insect evidence at the crime scene, you want to first just observe what is present. Document the location, the environment, what insects you see around the corpse, what species, and where they're located on the body. The next step would be to collect meteorological data. You want to know the temperature of the body and the environment. What is the soil consistency underneath, because that can tell you what kind of insects will likely be there and how rapidly a body would decay. And you want to know recent weather events. Did it rain while the body's there? What's the humidity? After that, you can then begin collecting insects, and you want to do 2 different collections. The first collection is to collect any adult insects you find, alive and dead, and then collect the youth, the juveniles, the larvae, and the eggs. When doing a collection, you want to capture your insects with a net and place them in what's called a killing jar. It's a jar filled with 75 to 80% alcohol, and date and label the jar on when you collected it and what you think it is. When dealing with live insects, you want to keep them in a small container with some holes, soil, and food to make sure they're alive by the time they reach the lab. The last step is, after the body's been removed, look for any surrounding insects. Perhaps there were insects underneath the body that weren't initially captured during your first 3 steps. Here's just an overview of what that would look like. When analyzing a body, you want to always look for where are the insects, probably going to tell you where injuries are. You're going to want to use nets, killing jars, and you're going to want to be very mindful of the areas surrounding the body. So we started this with a case. We had found a body in the woods. It appeared bloated and smelled really bad and had small maggot-like insects. Could you now determine how long that body was probably there? My hope is that you can. I hope this was helpful in your understanding of forensic entomology, and I'll see you next time.